Good afternoon, everyone. I would like to welcome you to our session on late tumor recurrence and tumor dormancy. We're in treat for 3 fantastic talks. We're going to start with 2 basic science talks and then have our third lecture be about the clinical implications of tumor dormancy. So first I should introduce myself, I guess. I'm Alana Whelm. I'm Senior Director of Basic Science at the Huntsman Cancer Institute at the University of Utah and Professor of Oncological Sciences there. We will have 5 minutes question and answer at the end of each presentation, so please come up to the mics if you have questions. I will at this time introduce our first speaker, who is Dr. Isaac Chan from UT Southwestern Medical Center. Dr. Chan is an assistant professor in internal medicine at UT Southwestern and a member of the Division of Hematology and Oncology. He specializes in immunotherapy, breast cancer, and metastatic cancer. Dr. Chan earned both his doctoral degree in genetics and molecular signaling and his medical degree at University of North Carolina at Chapel Hill. He did a residency in internal medicine at Boston University and then had advanced training through a medical oncology fellowship at Johns Hopkins University School of Medicine. He joined UT Southwestern in 2020, and his research, as I mentioned, is really focused on how to understand and treat metastatic disease by looking at the processes by which cancer cells spread, and today we'll hear about his work on deciphering the role of natural killer cells in breast cancer. So, welcome, Dr. Chan.
Thank you, Dr. Whelm, for that wonderful introduction, and I want to thank the organizers of the conference for inviting me to give this talk today. And so today I'll talk about deciphering the role of natural killer cells in breast cancer and breast cancer dormancy. I have no financial disclosures to report. And so the outline of today's talk, I'm going to talk about natural killer cells and their role in dormancy, and then what's their role in metastatic outgrowth, so the period after dormancy. And then we're going to talk about some of our own lab systems biology approaches to NK cell biology in the breast tumor microenvironment. But first, I just wanted to introduce NK cells a little bit to everyone and talk about some of their unique features. So NK cells are innate lymphoid cells that have potent anti-tumor and anti-metastatic properties. And they're first described in the 1970s in a series of papers that describe a cytotoxic cell that was very curious because it lacked properties of T cells or B cells. And so they called it initially the null cell. And then, after some more investigation, they found that the initiation of their cytotoxic function did not really require prior exposure to tumor antigens. And so that's what led to his moniker, the natural killer cell. And natural killer cells are regulated by activating and inactivating receptors. And so here are some activating receptors in the cartoon on the right. And so NKG2D is an activating receptor. And then there's some inactivating receptors that are unique to NK cells, like KLRG1 or NKG2A, and the KIRs. And they also share some inactivating receptors with T cells, such as PD1, CTLA4, LAG3, and TIM3. But eventually, NK cells lose their control of cancer cells. So even though they have very potent ability to eliminate cancer cells in the primary tumor and distant metastatic sites, every clinically significant tumor in a patient has somehow evaded this control. And so on the right panel, you can see a historical study from 1983 where NK cells were actually taken from healthy donors in the black bar, patients with early stage breast cancer in the white bar, and then in the stripe bar, they took NK cells from patients with metastatic breast cancer. And they compared their cytotoxicity or their ability to kill cancer cells, and they found that patients with NK cells taken from patients with metastatic breast cancer had the worst ability to kill cancer cells. So that really hints at what is going on in the metastatic setting, how are they evading, how are these NK cells being changed by metastatic breast cancer cells. And so dormancy might give us an answer and a mode of immune escape. And so dormant cancer cells evade immunity. They are defined as cancer cells that are non-proliferating and have undergone G0 to G1 cell cycle arrest. Dormant cancer cells have managed to avoid immune clearance, but the timing and the trigger of dormancy to metastatic outgrowth is unclear. And further, there needs to be an understanding or additional understanding of the impact of tissue-specific microenvironments on cancer cell dormancy. And so the studies that I'm about to highlight start to try to answer some of these questions. And so one study by Malati et al. Used cell line models of dormant cancer cells, and they engrafted them in athymic mice, so mice that have functional NK cells but dysfunctional T cells and B cells. And they compared it to engraftment of dormant cancer cells in immunocompromised mice, NSG mice, with no NK cells. And what they found was that there was decreased metastatic outgrowth following engraftment in athymic mice. Further, when they performed NK cell depletion studies using various antibodies, they found that the depletion studies resulted in increased metastatic burden from the dormant cancer cells. So this further suggested the role of NK cells in controlling metastatic outgrowth. So then they analyzed dormant cancer cells, and they found that these dormant cancer cells showed decreased gene expression of NK cell activating ligands and functionally had less NK cell-induced cytotoxicity. Further, what they eventually found was that dormant cancer cells can downregulate the Wnt pathway activity in order to escape proliferation, and it correlates with the expression of a Wnt inhibitor, DKK1. And this autocrine signaling permits the dormant cancer cells to remain in a quiescent state. And what they found was that when they knocked down DKK1, it increased sensitivity to NK cell cytotoxicity. And so this is the model they proposed, is that metastatic cells, when they arrive in the distant site, they're cleared. There's a lot of attrition. But some dormant cancer cells survive and express DKK1, which inhibits Wnt signaling, and that keeps them in a dormant state. But some cells are allowed to proliferate, and those are cleared by NK cell-mediated cytotoxicity. However, the dormant cells continue to express DKK1 and actively inhibit NK cell cytotoxicity, and that permits future metastatic outgrowth. So then this idea of timing and the period in between metastatic latency kind of suggests that there might be some differences in NK cell surveillance. And so the same group then switched to a brain metastasis model where they wanted to understand the biology underlying the differences between synchronous brain metastases, so brain metastases that appear with the primary tumor, or metachronous brain metastases where the primary tumor is treated and then metastases form later on. And they wanted to do this because this could inform clinical treatment strategies. And so Parita et al. Used HCC 1954, which is a HER2-positive cell line, as a model, and they performed an in vivo screen and generated cell lines from mouse brain metastases that reflected each phenotype. So essentially, they created cell lines that reflected synchronous brain metastases, metachronous brain metastases, and then dormant cells that did not form brain metastases. And then they performed metabolic analyses between all the groups. And what they found was that metachronous brain metastases secretes and uses the highest amount of lactate versus other types of brain mets. And instead, what they found was that when they lactate supplemented mice systemically, it could trigger overt metastatic outgrowth from dormant and grafted mice, and it also suppresses NK cell killing in vitro studies. So, we kind of have moved from different sites of metastasis, the properties of dormant cells and their ability to escape NK cell killing through different types of organs. And so this study by Correa et al. I thought was really interesting because it started to answer, are there organ-specific NK cell responses to dormancy? And so here, they had a unique model to track dormant cells. So they had a M. venus reporter of quiescent dormant cells, and they performed engraftment in mice and then harvested all the distant metastatic sites and the dormant cancer cells at those metastatic sites and also the surrounding stroma. And what they found was that the liver had the highest amount, or the highest percentage of dormant cancer cells. And when they analyzed the stroma around the liver, or around these dormant cancer cells, they found that the liver stroma expresses genes highly related to NK cells and have high amounts of NK cells. And very interestingly, in a series of experiments, they found that NK cells actually secrete interferon gamma to sustain dormancy. So it actually aids the dormant phenotype. So NK cells can aid the dormant phenotype. Further, they noticed that the liver microenvironment is very unique and contains lots of hepatic stellate cells, or myofibroblasts. And they found that these myofibroblasts in the liver were activated and highly elevated around metastatic liver lesions, but not activated around dormant cancer cells. And so what they did was they used a carbon-TET liver injury model, which is a classic experimental model of liver injury, and they combined it with their metastasis model to induce the numbers of activated hepatic stellate cells. And then they analyzed the NK cells. And what they found was that NK cells are decreased in these models, and they're actually kept in quiescence. And that's what permits metastatic outgrowth. And so the model they propose is that these dormant cancer cells arrive, and then they're actually kept in dormancy by secreted interferon gamma by NK cells responding to those dormant cancer cells. But as they start to proliferate, they create liver injury, and they activate hepatic stellate cells in the liver microenvironment. And those hepatic stellate cells then suppress NK cell activity by putting them into quiescence. And so these fascinating studies really lead to a lot of outstanding questions. What are some of the other signals used by dormant cancer cells to suppress immune response? Or do they even activate other immunosuppressive cells like MDSCs? How do other aspects of the tissue microenvironment signal to NK cells during dormancy? How do we capture all the different aspects of the tumor microenvironment and their role in suppressing or engaging NK cells? What is the role of immune cell quiescence in controlling or failing to control metastatic outgrowth? Another additional question is, it was so fascinating that these NK cells can secrete interferon gamma to sustain the dormant phenotype. Are NK cells supportive of cancer cells in general? And so that's a question that my group tried to answer a little bit. And so we actually started a different approach to all of this. We wanted to understand which immune cells respond to metastatic colonies. And so we performed this experiment where we took tumor clusters from PYMT mice and grafted them into wild-type immunocompetent mice, and then at the end of 6 hours, not the usual 2 months, assessed for the innate immune response at the lung. And what we found was that maybe there are about five on average, 5 macrophages around each red and green tumor cluster, maybe 2 to 3 neutrophils. But then we saw, on average, 15 NK cells and as many as 28 NK cells surrounding each one of these metastatic clusters. And so that really tuned our attention that NK cells could be important in this process. Now, I just want to add that our knowledge of NK cells have historically come from established tumors. And so that's why you might hear, NK cells form about 1% to 6% of an established tumor. But it doesn't necessarily capture the response of the NK cell right as the tumors hit and colonize a distant metastatic site, where they're about 70% of the innate immune response. And so we created an organoid-based platform to study NK cell and cancer cell interactions. And what we essentially do in brief is we take tumor tissue from human breast tumors or mouse tumors, and we perform a series of enzymatic digestion and differential centrifugation steps to arrive with enriched epithelial organoids. And then we embed those organoids in different types of extracellular matrix. And then we isolate primary NK cells from human or mouse, and we put them in co-culture with our organoids. And I'll just quickly highlight some of the advantages of our platform. This 3D culture in different types of ECM allows us to model specific cell-cell interactions along with morphologic, spatial, and temporal features of immune and tumor cells ex vivo that simply aren't accessible in vivo. And I'll show you a couple movies of what I mean. The platform is very adaptable and scalable and can be quickly assembled within 4 hours, as opposed to 2 weeks, to test therapeutic, functional, and molecular phenotypes of immune and tumor populations. And in contrast to other isolation methods, our organoid isolation method captures intratumoral genetic heterogeneity. And that's a topic for another time. So I want to see if these movies will work. I was told that they might not work. Oh, no, that works. So this is what I mean by our platform. So on the right, you can see an organoid that's free to invade into the extracellular matrix. And on the left, you can see that. And on the right, when you add NK cells into the co-culture platform, you can see their ability to restrict the metastatic invasion. And so this is our model of invasion out of the primary tumor. But this platform can also capture spatial and functional information at the single cell level. So here is a movie taken every minute over 12 hours of NK cells interacting in red, interacting with blue tumor organoids. And there's a Cas-based molecular reporter in the system. And so you can then imagine the molecular reporting we can do to capture functional information as each NK cell engages those tumor organoids. And so we used this platform to actually understand a little bit more about NK cell biology, and we found that NK cells initially are able to restrict metastatic behavior, but then by targeting these keratin 14 positive MHC class 1 negative metastatic tumor cells, but over time they can actually be reprogrammed or corrupted to promote metastatic outgrowth. So similar to this theme, that NK cells can be converted into tumor supporters. And this is just a summary slide of several experiments that we did to show that the reprogrammed NK cell can actually promote metastatic outgrowth in both our in vitro models and then our in vivo models, both increasing the number of metastatic colonies formed. And so with the remaining time, I just wanted to talk a little bit about our systems biology approaches to understand NK cells in the breast tumor microenvironment. And this is actually quite a theme that's in San Antonio. How do we make sense of all the big data that we have? How do we use that to generate better and more accurate information about the breast tumor microenvironment? And so my students, Lily and Zhu and Caitlin Saunders, have been working on this project. And Lily's actually giving a general session talk on this very topic in Hall 3 on Friday, December 9th, from 1:15 to 1:30 p.m. So I just wanted to put that plug in there. She's going to be talking about this in depth. But essentially what we did was we created a large single-cell RNA-seq data set of breast tumors. It contains over 119 samples, over 236,000 cells. And that allows us to be powered to actually start interrogating rarer immune subtypes, like NK cells here in this cluster. And so when we isolated the NK cells out, we actually were able to capture 6 clusters of NK cells. And so cluster NK0, NK2, and NK4 were more cytotoxic. NK3 were more representative of tissue-resident NK cells. And NK1 and NK5 were representative of inactive NK cells. And NK1 is pretty special, and I'll tell you why in a few slides. But I first wanted to show this graph here, this bar plot here, where each column is a patient and is sorted by clinical subtype. And it actually just highlights the tremendous diversity and heterogeneity of NK cells within each person, despite their subtype. And so it starts to hint at our ability to maybe one day be able to identify patients' immune profiles and use that as a way to guide therapy. But one of the questions that we had was, can we translate our preclinical findings on reprogrammed NK cells to the clinical arena? And so we wanted to generate a gene signature of reprogrammed NK cells and apply it to our data set. And when we did that, we found that NK1 in an unbiased fashion highly expressed the reprogrammed NK cell signature above all the other clusters. And they also shared very similar genes to NK1, which was NR4A family, DUSP1, FOSB, and Jun. Further, patients in the TCGA with a high expression of reprogrammed NK cell signature correlated with worse survival outcomes over 10 years compared to those that have a low expression of reprogrammed NK cells. And we're able to perform some receptor ligand pairing to capture receptor ligand interactions between different subtypes of cancer epithelial cells and reprogrammed NK cell receptors. But I'll quickly just introduce this concept because Lily is going to talk about this in detail. But essentially, another theme of this conference has been the tremendous amount of cancer epithelial cell heterogeneity within the breast tumor microenvironment. And as a result of that, we wanted to understand how our immune interactions change by this heterogeneity. And so what we did was we actually took all the cancer cells in our data set, and perform unsupervised and supervised clustering to categorize all those cancer cells in 10 categories, or gene elements. And so by using these 10 gene elements, we were able to define all the different types of cancer, of the heterogeneity of cancer epithelial cells. And then we used the decoder to predict the cancer cell and immune cell interactions based on the GE expression on the x-axis. And so just to summarize really quickly, heterogeneous breast cancer cells can be defined into 10 GEs. We can predict immune interactions for each GE. And then we can also predict GEs that might interact with NK cells. And this can be validated experimentally and using spatial transcriptomics. And so one of the future directions is actually then understanding, maybe some of these GEs are a dormant NK cell, or dormant cancer cell expressing category. And I just want to end my talk with this provocative question of whether immunotherapy can be personalized. Can we one day take a person's tumor, deconstruct it into different components, and then identify which one of those components will respond or not respond to NK cells in this case, and then develop therapies, NK cell-directed therapies, for these specific patients. And so with that, I just wanted to end by thanking everyone in my lab, especially Lily and Caitlin, our collaborators, our funding sources, and especially patient advocates like Julia and Christine, who've really been equal partners in this and also have significantly advanced the research that we do. Thus showing that partnering with patients, I think, makes a significant impact on the research. And so I'll take any questions that we might have.
Thank you very much, Dr. Chan. We're just about out of time, but if there's time for one question, we can take one, if there's anybody who wants to come up. Or you can hold it till the end, and if we have more questions, if we have time at the end, we can take a question.